I'm pleased to present our last presenter for today. Will be Dr. Michael Hayden, CEO of Prilenia. Dr. Hayden will report on pridopidine for the treatment of ALS, reporting results from their phase 2 clinical trial, which is conducted through the Healy ALS platform trial. Welcome.
Good afternoon. Thank you, Fernando, and thank you to ALS ONE for giving me this opportunity to share progress and provide an update on pridopidine in ALS. And as a start, I did want to just thank ALS ONE for bringing us all together in this special form. I also want to express my appreciation, gratitude to all the patients across the United States that have embraced and participated in the Healy platform trial, in particular Regimen D that I'm going to present, the investigators, and of course my partners at MGH at the Healy Center and my colleagues at Prilenia. So I will just go to the presentation now, to presentation mode. I'm going to be talking to you about pridopidine for the treatment of ALS. Just to start, pridopidine is an oral molecule. It's a sigma-1 receptor agonist, and we know this from confirmed in vivo human studies. How these work is essentially you use a sigma-1 ligand that binds sigma-1 firstly to show occupancy, and here you can see beautiful occupancy lighting up with fluoxetine. Then you do another experiment, having shown that that actually locates at the sigma-1 receptor. Then you pre-treat with your drug pridopidine, and then after that you use fluoxetine, and you see how much of this is blocked. And what you can see here, it's almost completely blocked, telling you that this is a highly selective, potent sigma-1 agonist. We did the same thing, because initially this was thought to be acting on D2 and D3 receptors, and there was no diminution of signal. So this is a sigma-1 receptor, we now know agonist from functional studies, and primarily not a D2, D3 receptor. Now, there's further importance and evidence about sigma-1 genes playing a role in ALS. These are rare mutations that highlight the important role of the sigma-1 in motor neuron function. This comes from numerous genetic studies in humans that shows that if you have a mutation in sigma-1, you get ALS. And furthermore, what's most interesting is that when the mutation results in a null allele or no protein being produced, such as these here, you get a very severe juvenile form of ALS. And when it's a partial mutation, losing about 50% of function, you get adult onset ALS. So very strong genotype-phenotype correlation. Of course, this is no different to the LDL receptor for hypercholesterolemia that led to the statins. The question, of course, now becomes, well, what happens if you agonize sigma-1? Do you improve the function of motor neurons? And just to remind you about the mechanisms contributing in ALS and numerous other neurodegenerative disorders to neurodegeneration, these are the mechanisms that sigma-1 agonism really counteracts. There's increased ER stress that's a result of disturbance in calcium homeostasis, leading to decreased mitochondrial function, less contact between the crucial ER and mitochondria, leading to decreased autophagy, decreased synaptic function. Various neurotropes like BDNF are decreased in secretion, and you get loss of connectivity in spines. And what pridopidine does in ALS and numerous other models for neurodegeneration is reverse every one of these mechanisms. We know it's sigma-1 receptor dependent because when these mechanisms are further assessed in animal models, say for ALS, where sigma-1 is knocked out or is not present, you lose the effect completely. So this is truly a sigma-1 receptor effect. Now, we went on to look also in human-induced pluripotent stem cells. These are derived from patients looking at models of ER stress and to see how pridopidine worked in these models that come from patients with ALS. And of course, prolonged ER stress is a key feature in ALS. And we know that in models, reducing ER stress increases cell survival. So here we won't spend too much time on this, but here's the model. Pridopidine, for example, here at 1 micromolar in the presence of the stressor tunicamycin dramatically reduces ER stress. And this is measuring BIP, which is a marker of ER stress. And what you can see, this BIP is dramatically reduced by 75% in these induced pluripotent stem cells. Also looking at cell death pathways, and CHOP is an important ER stress, upregulates CHOP, and this is associated with caspase pathways. And here you can see in the presence of just tunicamycin, you see it's normalized to tunicamycin. Again, you see a very nice decrease in CHOP associated with this. And finally, if you look at cell viability, in the presence of ER stress, here you can see cell viability just with the stress around 21%. And as you get to 1 micromolar pridopidine, you restore vitality of cells to a significant degree. So these are just models. This, of course, suggested that pridopidine can reduce ER stress, and furthermore supported studies that will assess pridopidine, which is a safe oral drug, in clinical trials. Just to also mention, there's long history of safety with this molecule, and the safety profile after, close to 1,600 patients, mostly Huntington disease, but also ALS, has a safety profile comparable to placebo. So let me share with you some of the phase 2 results from the Healy platforms trial. We were privileged to be amongst the first 4 in this platform trial. And you will recall that this trial shared placebo across 4 arms, and we were regimen D, so we had 120 patients in active, 40 in placebo, but placebo was shared amongst 4 arms, so there were 160 in these patients. Important to note, this was also to some extent an all-comers study where they had inclusion criteria of up to 3 years and a vital capacity of 50%. So this was including some patients who were already quite advanced. I'm going to show you the subgroup that we analyzed that will be part of our phase 3 study that we are planning and moving ahead on. This is the group that's definite, probable, and early. These were importantly because these are the patients that have progression in a fairly predictable fashion and would be expected on placebo to be decreased, so there might be a window that you could detect the effect of your drug. And what you can see here, just in this phase 2 study, we already saw a change, a disease slowing of progression, even by 8 weeks. And this was sustained all the way out to week 24. This was a 24-week study. Associated with this, when we look at forest plots, you can see the ALS FRSR is significant at each measure, and it's contributed by each of the components. Every component contributes, but it is interesting that respiratory and bulbar seem to be the major contributions, with combined motor assessment slightly less. Now, in respiration, it's interesting, and it's not totally unpredictable, because sigma-1 is most highly expressed in the brainstem, and the brainstem, as well as motor neurons, but the brainstem is very important in respiration and bulbar function, eating and swallowing. When you look at for example, just one parameter in the respiratory assessment, which is dyspnea, again, what you see very nicely is that the placebo, there's a window to see an effect, and there's minimal change up to 24 weeks in patients who are in these definite, probable, and early, showing the effects on the respiratory subscales. We also see the same in bulbar, where again, the difference from placebo is showing with really some slowing. Even with small groups, we are getting close to significance at 24 weeks, and certainly some slowing in bulbar function at 16 weeks. When we look at bulbar function again, you can see that each of the parameters contributes to this: swallowing, salivation, and speech. And we did in this study look specifically at speech using the oral analytic breakthrough designated device. And here you see again, very impressively, slowing of decline in articulation rate and slowing of decline in speaking rate compared to baseline, all compatible with improvement in bulbar function. Now, I'll just show you with these changes in respiration, we were, of course, very interested to see the changes in survival. Just to say, at 180 days, the end of the double-blind process, there were 4 in placebo, 3 in treated, so really not enough time to have enough to see an effect. After that, everybody went on to predopidine. So these are the delayed start, and these are the persistence. And what you can already see is some meaningful separation between a delayed start and a longer-term treatment effect with predopidine over the delayed start. When you look at it, one measure for survival is, of course, 50% survival probability. In the delayed start, this was around 50%. At that particular time, the patients who had been on predopidine from the beginning of the study, it was only 15%. The question became is, when do they reach 50% survival probability? And that was about doubling of survival compared to the late start, and this was about 10 months later. So there appeared to be, although small numbers, but this was really sufficient to give us some confidence to be able to want to examine this in a longer-term study. We did work with Berry consultants who did some initial analyses looking at the PROACT database. And this is where there was information on patients where we knew they had died. So there's a bias in favor of predopidine just by the virtue of the fact now we know these patients have died. But when you looked at this and you look at 50% survivability, this is also increased in this group and with some nominal significance to PROACT. So we were approached by Healy at this point, who were also encouraged by these findings to apply with to the Act for ALS to get an NIH-funded grant. This was submitted and funded by the NIH, where we got the notice in October. The first patient, and this is allowing patients to have expanded access of this drug now, but these patients will have greater than 24 months duration, which is going to be outside the bounds of our phase 3 study. I must say it started in April, just end of April. It's just been a short period. We are recruiting 200. Recruitment has been rather rapid. 110 patients recruited at 25 sites, and this we hope to continue with the recruitment with up to 200 patients by early 2025. Recruitment has been outstanding for this study. So in summary, I'll show our analyses in these patients, the early and these fast progressors, show significant changes in ALS FRS total, respiratory, dyspnea, and bulbar. And these pre-specified analyses also showed quantitative speech improvement. Our survival analysis gave us encouraging benefits, and no safety signals were identified. As we know, this is a very, very safe product in a large number of patient years. And so these observations are informing our planning now for our phase 3 study. And just to give you some perspective, of course, we want to be able to have mortality as part of this. This will be a 48-week study with definite probable and less than 18 months. SVC will be up greater than 60%. This is going to be a 2-to-1 randomization, double-blind, placebo-controlled study for 48 weeks with 48 weeks extension. And some of our clear secondary endpoints will look at speech, of course, respiratory function, quality of life as measured by the ALS-AQ40, and survival which is going to be important as part of this. This will be conducted in Europe, as well as the United States and Canada, and we are looking forward to showing this. Let me just show you one other story that may provide just another perspective about the neuroprotective effects of pridopidine. As many of these neurologists listening will know, vanishing white matter disease is a genetic disease, very serious, usually leading to onset, usually in the first 15 years of life, leading to mortality fairly rapidly. And we, together with Paul Tisar at Cleveland, were approached by Mass General because in animal models, pridopidine had shown some effects, with sigma-1 being the target. So what Paul did is he created organoids from patients from a disease, and these were, one of the subjects had this particular mutation, homozygous for this mutation. What you can see here, and created organoids associated with this, and what you can see here is these organoids have decreased oligodendrocytes. Here you can see, here's the control, very significant decrease in oligodendrocytes. And the question is, what would pridopidine do to those? And what you can see here is pridopidine had big effect in enhancing survival compared to the homozygote with this, improving the survival of these oligodendrocytes. Then Dr. Florian Eichler, who's been has been the investigator on this study, has just reported on these two patients in June this year, where this was an intermediate-size expanded access protocol. These participants were 22 and 39. They took twice-daily pridopidine. And what is remarkable and unexpected is that these patients remain totally stable with no changes in assessment scores, and CT and MRI, again, showed no further progression. And I'll just show you this. NFL levels were measured in these patients. NFL levels go up with age. So at age 20 to 29, the mean would be 4.4. Then the 30 to 39, a little higher. And what you can see here is this patient started off around 8. And then by week 96, very close to what would be expected in terms of decrease of neurofilament. Subject 2, a little lower, a little older, also had a nice decrease in neurofilament up to week 48. So these are patients that had some stability in terms of their clinical progression, confirmed by various assessments of cognitive function. MRI and CT also showed stability, and here we saw a very nice decrease in NFL. And of course, NFL will be part of our study going forward. So I just want to thank you for giving me the opportunity to share some of this encouraging data with you. And of course, we'll be coming back to the community to talk further about our phase 3 study in the near future. Thank you very much.
Thank you, Michael, for the great presentation and describing what will be a really robust clinical trial. That phase 3 clinical trial plan is incredible. There are a couple of questions in the chat. So one question asks, does pridopidine stimulate oligogenesis? Is that a concept that you're exploring?
Well, pridopidine has what we see in vanishing white matter. So we haven't done with the iPSCs really assessing that. We assess some of the ER stress and the pathways associated with that. But what you do see is that pridopidine, in all likelihood, does stimulate oligodendrosis genesis, because we see in these patients who you saw in that slide, and this is Paul Tisar's work, where we saw the patients come in with these organoids have decreased essentially oligodendrocytes, and then you see a very significant increase after treatment with pridopidine. So Paul has really assessed that in those models, and I think he would say that that's likely, but it hasn't been tested in iPSCs at this point.
Thank you. And I just had another question. You focused a lot on the mechanism of action around ER stress. and EF2 beta activation in particular, how do you imagine pridopidine might differ in its activity vis-a-vis other drugs that are going right at that target that are putative EF2 beta activators?
Well, pridopidine has impact, I think, a little bit more globally. So it does stimulate EF2, you know, what is similar to the Denali compound in terms of activation, and they're also hoping to do a study in vanishing white matter. But the pridopidine has much broader effects. Pridopidine has impact on BDNF, which is so important for neuronal effects. Pridopidine restores calcium homeostasis. Pridopidine has impact on mitochondria directly, and can impact on mitochondrial respiration. Pridopidine also enhances the contact between. So pridopidine is like a hub. It's a central hub and acts as a molecular chaperone, which then interacts with many different targets. IP3 receptor. Pridopidine actually binds the IP3 receptor to restore calcium homeostasis in the mitochondria. And that calcium homeostasis and the ER is so important in terms of the contact with the mitochondria to enhance mitochondrial function. So both are really important, but I would suspect pridopidine has broader effects around multiple mechanisms by acting as a molecular chaperone, interacting with numerous targets as part of its effects. We also know, of course, that the data from sigma-1 human genetic mutations, where patients with sigma-1 essentially develop ALS, you know, the most direct link between sigma-1 and ALS, and we know that it's an agonist. For example, pridopidine has big effects on BDNF secretion and numerous other neurotropes. So the effects would be more generalized in terms of neuroprotective pathways.
Thank you. And being respectful of everybody's time, we'll do one more question that two folks expressed. They ask, do you plan to combine with SOC? Any evidence of synergy?
Well, at the moment, we're really looking at pridopidine alone. We, of course, will be going with standard of care, such as riluzole and edaravone. These would have particularly for edaravone, randomized at baseline, so there'd be balanced randomization that pridopidine, you know, there'd be equal numbers in both groups. Riluzole, we saw in our last study, 75% of patients were taking riluzole. When we looked at patients with pridopidine and riluzole, we saw no change in the effect. With edaravone, it's still under discussion, but there might be some additive effect with edaravone, and so we will do balanced randomization of edaravone at baseline.
Thank you very much. And with that, we'll close today's industry day of the 7th annual ALS ONE Symposium. I'll turn it over to Jen